Protoplast fusion is an important tool of gene manipulation because it breaks down the barriers to genetic exchange imposed by conventional mating systems. In nature, plants from far related or unrelated species are unable to reproduce sexually as their genomes or modes of reproduction are incompatible. A pollen grain attached to a stigma does not succeed in forming a tube or delivering the sperm cells because interspecific barriers prevent fertilization between different species. This natural barrier is met over by the ability of these naked cells to fuse with each other irrespective of their origin. Hence, protoplasts from unrelated species are fused to produce interspecific or even intergeneric hybrid plants combining desirable characteristics such as disease resistance, good flavor, cold tolerance, etc. This process of raising novel hybrids through the fusion of somatic cells bypassing sex altogether is known as somatic hybridization. For protoplast fusion, physical contact of two protoplasts is essential. Protoplasts do not fuse easily due to two main reasons. They have a net negative charge on their membrane surface due to intramembranous phosphate groups, and force of repulsion works between protoplasts. And it is difficult to remove water from hydrophilic surfaces of protoplasts, which also create a repulsive force between two protoplasts. Hence, the methods for fusion were designed to counter such hurdles. Three important methods are generally used for protoplast fusion. First one is high calcium and high pH. Second is polyethylene glycol fusion. Third is electrofusion. High calcium and high pH induced protoplast fusion. It was observed that positively charged ions reduce the net negative charges of membranes, reducing the repulsive forces considerably. Keller and Melcher in 1973 found calcium ions suitable for such purpose and developed this method of protoplast fusion. They fuse protoplasts of two lines of tobacco using high concentration of calcium ions at alkaline pH, incubated at 37 degrees centigrade for about 30 minutes. This method works well, but for some plant systems, high pH may be toxic. Hence, other fusogens are also used. Polyethylene glycol induced fusion. PEG induced protoplast fusion was developed by Kao and Michailuk in 1974. This technique is relatively more efficient. Since the polyethylene glycol molecules have polarity like membrane phospholipids, they get attached with membrane proteins, and when the attached PEG between two protoplasts is removed, it results in breakdown of membranes at the contact points, causing protoplasts to fuse with subsequent rejoining of plasma membranes of the adjacent protoplasts. Several factors affect protoplast fusion by PEG. PEG molecular weight higher than 1000 induces tight adhesion and high frequency fusion of protoplasts. Generally, PEG of molecular weight 1500 to 6000 has been used at concentrations ranging from 15 to 45%. PEG induced fusion is enhanced by enriching the PEG solution with calcium ions. Protoplasts from young leaves and fast growing calli give better fusion. The protoplasts from cultured cells can tolerate enzymes, PEG and high pH, high calcium treatments fairly well, and those from the mesophyll cells have proved sensitive to these conditions. However, pre-culturing the leaves for a few days improves the tolerance of mesophyll protoplasts to these treatments. Protoplast density also influences the fusion frequency. A 4 to 5% protoplast suspension usually gives the highest frequency of heterokaryon formation. Electrofusion or electric field induced fusion. Electrofusion of protoplasts was first demonstrated by Senda et al. In 1979. Later, in 1982, Zimmermann and his co-workers developed this method further. In this method, protoplasts are placed in an electric field and are exposed to high intensity electric pulse for a short duration. This exposure to electric field increases permeability of the cell membrane. Local electrical charge breakdown occurs in the plasma membranes, resulting in fusion of adjacent protoplasts. The original properties of membrane are restored within microsecond to minutes, depending on the experimental conditions and membrane properties. The process of electric field induced protoplast fusion can be explained in 5 steps: dielectrophoresis, mutual dielectrophoresis, membrane contact, electric breakdown of membrane, and protoplast fusion. Close membrane contact of two protoplasts is one of the prerequisites for protoplast fusion. This step is achieved by dielectrophoresis. The particles become polarized in the presence of non-uniform electric field and moves in linear motion towards the region of highest field intensity. The protoplast moving towards the region of highest field intensity will approach another polarized protoplast during the movement. This will lead to the formation of chain-like aggregates with point-to-point membrane contact. This effect is termed as mutual dielectrophoresis. The attraction forces arising from dipole generation within protoplasts overcome both the electrostatic repulsive forces between membrane surfaces of neighboring protoplasts bearing net negative charges and repulsive hydration forces. The optimum conditions for the electric field induced fusion process are achieved when the membranes of the two adjacent cells flatten out on coming into contact with each other, thus forming a relatively large zone of contact. Once the exchange of materials from the zone of contact between the protoplasts takes place, the fusion is attained. Advantages of electrically induced protoplast fusion. The fusion process between any two protoplasts of different species can be followed under microscope. Fusion process is synchronous and extends over a short duration, so the hybrids do not lose the viability. Viability can also be affected by fusogenic compounds which interact with total membrane surface in an uncontrolled manner. In this method, there is no use of such fusogenic compounds. The yield of somatic hybrid is very high, and during the fusion, loss of intercellular substances is generally found to be very low. The heterokaryons, which are the potential source of future hybrids, constitute a very small proportion of the mixture. Either unfused protoplasts of the two species or strains or products of fusion between two or more protoplasts of the same species, that is homokaryons. It is therefore of key importance in somatic hybridization to select the hybrid cells or heterokaryons to raise a novel hybrid. Therefore, an effective strategy has to be employed for the heterokaryon selection. Morphophysiological basis. The whole mixture of the protoplasts is cultured after fusion treatment, and the resulting calli or regenerants are screened for their hybrid characteristics. In the cross Solanum tuberosum and Solanum acerifolium, one of the parents, that is Solanum tuberosum, produces bright green callus, and the other parent forms brown yellow callus with purple colored cells. The putative hybrid calli were identified by their intermediate morphology, that is green with purple colored cells. Complementation. In this case, complementation or genetic or metabolic deficiencies of the two fusion partners are utilized to select the hybrid component. Complementation methods depend on fusion of two protoplast systems, each of which carries different recessive selectable markers. The resulting somatic hybrid cells are functionally restored. For example, complementation of cytoplasmic albino trait of one parent, that is Petunia hybrida, and sensitivity to culture medium of the other parent, that is Petunia parodi. In these combination, when the protoplasts were plated in MS medium after the fusion treatment, Petunia parodi protoplasts were eliminated at the small colony stage. Only the protoplasts of Petunia hybrida parent and the hybrid component developed into full callus. The calli of the hybrid nature could be clearly distinguished from the parental type tissue by their green color, and Petunia hybrida callus can be easily identified and rejected by their albino callus. Mason et al. In 1989 and Thomas et al. In 1990 utilized the transformation of fusion parents, one with hygromycin and the other with kanamycin resistance genes, and heterokaryons were selected by adding both the antibiotics in the culture medium. Isolation of heterokaryons or hybrid cells. The manual or electronic isolation of heterokaryons or hybrid cells is the most reliable method. Manual isolation requires two parental type protoplasts which have distinct morphological markers and are easily distinguishable. For example, fusion of green vacuolated mesophyll protoplasts from one parent and richly cytoplasmic non-green protoplasts from cultured cells of another parent. In another case, the protoplasts labeled green by treatment with fluorescein diacetate are fused with protoplasts emitting a red fluorescence from exogenously applied rhodamine isothiocyanate. The dual fluorescence method helps in easy identification of fusion products. Any of these methods can be employed for selection of heterokaryon, but further proof must be added from other independent markers to finally prove or disprove the hybrid nature of the selected putative hybrids. Verification of selected hybrid can be done by morphology, comparison of number and morphology of chromosomes, or the banding pattern displaying isozymes from both parents, or demonstration of the presence of DNA from both parents through RAPD provides the most direct proof of successful hybrid. Types of somatic hybrids. Symmetric hybrid. Symmetric hybrid is defined as the combination of nuclear and cytoplasmic genetic information from both parental species. Since most of the hybrids obtained through symmetric protoplast fusion may contain numerous undesirable genes, repeated backcrossing and selection are required for elimination of undesirable traits. The most important application of somatic hybridization has been the building of novel germplasm as a source of elite breeding parents for interploid crosses and for crosses at the tetraploid level. However, the introduction of large amounts of exogenous genetic material along with the genes of interest may induce genetic imbalance, which may present undesirable characteristics which limits their utilization. Asymmetric hybrid. Asymmetric hybrid is incomplete with the loss of some cytoplasmic or nuclear DNA of one parental species during the mitotic divisions. Such hybrid is called asymmetric hybrid. Asymmetric hybrids may arise from spontaneous chromosome elimination in some distant combinations, but special treatments can be applied to increase the amount of genome elimination. This may be done by X-ray or gamma radiation, which induces chromosome fragmentation prior to fusion with receptor cells. The ionizing radiation can cause fragmentation of DNA and induces both single and double strand breakage, which leads to elimination of donor chromosomes, formation of microchromosomes and translocations. Asymmetric hybrids have been produced between Arabidopsis thaliana and Brassica napus and rice and Zizania latifolia. Cybrids. Cybrids harbor only one potential nuclear genome and either the cytoplasmic genome of the other parent or that of a combination of the parental species. Cybridization is used to transfer economically important traits such as cytoplasmic male sterility between species and to create de novo cytoplasmic male sterility systems between unrelated species. The cytoplasmic donor is treated with X-ray radiation to eliminate nuclear DNA. Subsequent fusion of differentially treated protoplasts result in the production of cybrids. Cybridization can replace the cytoplasm of a cultivar in one step, saving considerable time over lengthy back-crossing programs used to introduce alien cytoplasm into crops. Asymmetric hybrids and cybrids are widely used for crop improvement because one or more traits can be added while maintaining cultivar integrity. Novel interspecific and intergeneric crosses which are difficult to produce conventional methods can be easily obtained. Important characters such as resistance to diseases, ability to undergo abiotic stress and other quality characters can be obtained in hybrid plant by the fusion of protoplasts. Protoplasts of sexually sterile haploid, triploid, aneuploid plants can be fused to obtain fertile diploids and polyploids. Mitochondria of one species can be combined with chloroplasts of another species. This may be very important in some cases and is not achievable by sexual means even between easily crossable species. Techniques for protoplast isolation, culture and fusion are not available for many important crop species like many cereals and pulses. In many cases, chromosome elimination occurs from somatic hybrids, leading to asymmetric hybrid. Such hybrid may be useful, but there is no control on chromosome elimination. Many somatic hybrids show genetic instability, which may be an inherent feature of some species combinations. Many somatic hybrids either do not regenerate or give rise to sterile regenerates. Such hybrids are useless for crop improvement. Somatic hybridization technique is applied to increase the capacity of plants to cope better with abiotic and biotic stresses, which may become a key focus in the future for sustainable agriculture. These techniques can be applied to improve plants for ornamental purpose and nutritional quality, creating novel, highly desirable plants that can meet market demand.